Development and structure of pollen grains. Within the anther, there is presence of 4 microsporangia. Within each microsporangium, there is presence of sporogenous tissue which is centrally located. Sporogenous tissue consists of a homogeneous cells called pollen mother cells. Each pollen mother cell can undergo meiosis and produces microspores by the process called microsporogenesis. Initially, 4 microspores present together and it is called microspore tetrad. Later, each microspore will develop into pollen grain by vacuolation and one mitotic division, and this have one vegetative cell and one generative cell. Pollen grain represent male gametophyte which is nearly circular in shape and have the diameter of 25 to 50 micrometer. Each pollen grain consists of 2 layers which are protective in nature, exine and intine, and contain 2 cells, namely vegetative cell and generative cell. Exine is the outermost layer, consists of a chemical called sporopollenin. Sporopollenin is the most resistant organic substance present on earth because it is resistant to high temperature and it is resistant to strong acids and alkali, and there are no enzymes found yet which can digest sporopollenin. And because of this exine and sporopollenin, pollen grains are well preserved as fossils. Exine of pollen grain exhibit various designs and pattern. So those diagrams are given in the text. Germ pore is the prominent aperture on the exine where the sporopollenin is absent, through which pollen tubes comes out of the pollen grain. Intine is the inner continuous layer made up of chemical cellulose and pectin, and pollen tube emerges from the intine. Vegetative cell is large and have dense cytoplasm and irregular shaped nucleus with abundant food reserves, and the function of it is to give nourishment to the generative cell and produces pollen tube. Generative cell is the small and have dense cytoplasm and is spindle shaped. And very importantly, it is suspended in the cytoplasm of vegetative cell to get the nourishment, and it produces 2 male gametes upon mitosis. In 60% of angiosperms, pollen grains are released at 2-celled stage with 1 vegetative cell and 1 generative cell. After pollination, 2 male gametes are produced from 1 generative cell. In remaining 40% of angiosperms, pollen grains released at 3-celled stage, that is with 1 vegetative cell and 2 male gametes.